Presenting on Alzheimer's, and she's a first-time presenter, so what that means is you can ask clarification questions, but don't be mean.
Unless you're a teacher or Andrew Verdesco.
Isabella.
So the title of my paper is CABL Stabilizes HDAC Levels by Tyrosine Phosphorylation, Repressing Neuronal Gene Expression in Alzheimer's Disease. So in simple terms, that's a treatment to change gene expression in Alzheimer's. Okay, so why Alzheimer's? Alzheimer's disease is a neurodegenerative disease, which means it gets worse over time. And it affects your brain and the way you think. And it's basically the neurons and the synapses in your brain can't connect properly, so you can't send thoughts in the way a normal person would, a healthy person. And it can lead to memory loss, loss of motor skills, and just overall affects pretty much every part of your life. And it affects 4.5 million Americans right now, and it gets more common every year. Just as an example, if I say, where are we right now? I'm assuming pretty much everyone in this room can answer that question. But if you had Alzheimer's, you probably wouldn't be able to answer that question, or another simple question like that that we all just take for granted as immediately being able to answer.
Question.
Yeah.
Question.
Me?
Yeah.
Oh, no.
Oh.
Okay. So if you look at the picture of the brain, you can see basically how big of an effect this has on your whole brain. So the question with Alzheimer's is it genetic or is it environmental? Because until now, people have pretty much just been assuming that it's something that probably runs in your family and you just kind of have to wait and see if you develop it. What this paper is about and what the scientists are starting to figure out is that a large part of it is environmental, so it can come from what you've been exposed to throughout your life. So that's pretty important because if it comes from something that we're exposed to like in our childhood or in our teenage years, we could be exposing ourselves to things now that could lead to Alzheimer's later in life. Okay, so some terms to know before I start. Imatinib is a cancer drug and it inhibits CABL. CABL is a tyrosine kinase, which means it regulates protein levels, so it basically turns on and off the proteins. HDAC is histone deacetylase, and it's involved with the function of neurons, so it basically decides if a gene is going to be expressed or repressed. That's important in this because it's part of epigenetics, so it's how an outside factor could control your gene expression, and through your gene expression, you can just control your neuronal and synaptic function. So in Alzheimer's, patients have gene repression, which means that they have decreased neuronal and synaptic function and other symptoms of Alzheimer's. Okay, so before this started, the scientists already knew that high HDAC levels were linked to loss of neuronal function, so the Alzheimer's patients had high HDAC levels. They also knew that CABL was overactivated in Alzheimer's patients, and they wanted to know how these two processes were related or if they were related. So the first thing they did is they have imatinib, which is the cancer drug that inhibits CABL, which Alzheimer's patients have a high level of. So they gave Alzheimer's model mice imatinib to inhibit the CABL and try to see if it brought down the HDAC levels. And with the HDAC, it would bring down the symptoms. So if you look at the part that's boxed in red, ABO is what activates the CABL pathway. So this would be an untreated Alzheimer's patient. You can see how much HDAC they have. And you can see that with the imatinib, it basically brought their levels down to basically the same level as the control. And this graph is just showing that in a different form. Does anybody have any questions?
So the ABO would have forced the Alzheimer-like symptoms, or at least the production of the HDAC?
The ABL starts the CABL. I mean, the ABO starts the CABL.
Right.
Yeah.
Okay.
Question, yeah.
What's the
this is just imatinib, just testing. It's basically a control, just to see if you just gave imatinib, would there be any difference? Okay. So now they suspect that CABL is what's increasing the HDAC levels, but they want to confirm the relationship between the two. So they expressed CABL in hippocampal cells, and they saw that it led to higher HDAC protein levels. That's this graph. So you can see the control versus the CABL, and the CABL leads to higher levels of HDAC. And then this graph is showing when what's boxed in red is a CABL inhibitor, which is basically what imatinib is. So imatinib is in this graph. These two are an active CABL pathway with HDAC. And you can see that the activity levels are way lower, which means that more genes are being repressed, which would mean more symptoms of Alzheimer's. So when they gave the active CABL, when they inhibited that with imatinib, it brought the activity levels back up to normal, which means a normal amount of genes is being expressed, which is good because it would mean less neuronal damage. Any questions?
You showed a picture before where the brain of an Alzheimer's patient was significantly reduced in size.
Mm-hmm.
I mean, at some point, will imatinib not have any effect on that because of loss of physical neurons, or does it still have an effect?
They haven't tried it yet in humans, so I don't. Yeah.
What is KD?
KD is kinase 10, so that's why that CABL is inhibited, which makes it imatinib. Anything else?
Going off of like what Keisha said, would this drug kind of like help kind of regenerate neuronal cells, or would it just stop them from being inhibited or overexpressed?
I don't know if it could regenerate them. It's more that it's stopping like future genes from being repressed. I don't know. I could be wrong. Okay, so just to sum up what they've done, they gave imatinib to Alzheimer's mice models, and they saw that these two red boxes are comparing a wild-type mouse who did not have Alzheimer's and was not treated with anything with an Alzheimer's mice who was treated with imatinib, and you can see that it brought the level down to pretty much the same. So that's significant because it's a level that won't lead to gene repression and won't lead to symptoms of Alzheimer's. So the other graph is just a visual representation of the same thing. Can anyone explain the other graph to me? This one.
I think we're having trouble seeing it.
Okay, it's a little hard to
I'm going to
see.
shut off all the lights so that people can see it a little better.
Okay.
Yay!
Okay. It might be a little hard to see, but can anyone explain what that means to me? Okay, I'll go over it then. I can't see you.
It's actually nice. If I look at if you look at the imatinib levels, then it has less HDAC too than the levels of the one that had Alzheimer's.
Yeah, basically, the one on the left is a control group, so it had Alzheimer's, but it wasn't treated with anything. And this group was treated with imatinib, and you can see how much lower the levels of HDAC are in the group that was treated with imatinib, the red being the HDAC. Everybody good on that? Any questions? Yeah.
What's the difference between top and bottom?
They're just different parts of the brain that they tested in.
The hippocampus is responsible for memory.
Anyone else? So, what they learned was that imatinib, which is the cancer drug, can decrease HDAC levels in mice models of Alzheimer's. And they also learned that CABL directly regulates HDAC. So now they know this pathway, which means that CABL not only regulates HDAC, but regulates gene repression. And so if you inhibit it, you can prevent the symptoms of Alzheimer's. So, the next steps... Oh yeah, question.
How does
someone with Alzheimer's like first develop high levels of HDAC? Like how does that first?
Like what leads to?
Yeah.
That's one of the things that they're going to have to research. So the next steps for them would be to try to block CABL and the HDAC pathway in humans with imatinib. So something that's really cool about this is that imatinib is already an FDA-approved cancer treatment, and it's already in use with humans. So if they were to do trials in humans and it worked well, they already have this drug ready to go, and they know that it works in humans. So they would try to test and see if it works in blocking the symptoms the same way it does in mice. Yeah.
Is this one of those drugs where, like, if you take it, like, it will help reduce the symptoms at one point, but then if you stop taking it, the bad symptoms will come back?
Well, I imagine since it's stopping the high levels of HDAC, and if you stop taking it, I would imagine that the levels would continue.
Is that your question?
Yeah.
Yeah.
Like, are there like any other important functions of like the CABL pathway, like with blocking it, like any of the like side effects?
They didn't say any side effects. Yeah.
Can people build like a tolerance to this drug, like not when it comes to drugs and such? Or
I don't know. Sorry. Yeah.
Do you know the exact mechanism for how it stops HDAC too?
Stop. No, I don't know the exact name. Sorry.
You're getting.
I know I'm kidding.
You're getting the questions from the cancer class.
Okay.
So they're asking.
Yeah.
What would you do as cancer? Would it block CABL? Like,
If they use the job of imatinib is to block CABL, so it does the same thing. Yeah.
So you said imatinib was used as a cancer therapy. So why did they begin to investigate how it can block HDAC in Alzheimer's?
Because they already knew that in Alzheimer's patients, CABL was overactive, as well as HDAC, so they wanted to see if bringing it down would help with Alzheimer's patients. And the job of imatinib is to inhibit CABL. Yeah.
So if the mouse already has Alzheimer's and then it gets the drug, does it repair some of the neurons, or does it just prevent it from getting?
I think it would just prevent it from getting worse. Anyways. Yeah. That's good.
What kind of cancers does it have?
I want to say leukemia. No? No? What is it?
Out of the scope of your study.
Out of scope. Okay. No, it said it.
It was in your study?
Yeah. Or I googled it. What does it matter?
Okay.
I mean, I like should know that.
Alright, just be careful with this one. If you're not sure, don't answer.
No, I Google it. I'll get back to you.
Okay.
Okay. Okay. So. Okay. So not only can they try testing in humans, but they can also test with other neurodegenerative diseases that have the CABL pathway, which they already know this pathway is involved in other diseases, so they can see if this has the same effect in other diseases. And the other cool thing about this is that now they know this is the mechanism that an outside environmental force can use to turn off a gene and lead to Alzheimer's. So if they do some research into like what environmental factors are causing this, now they know that this is how they're doing it, but we can look into what are the factors that are causing this, and then we can basically see how to avoid them or how to deal with them in our lives. Any questions? Yeah.
How long were they treating the mice with imatinib for? What was the length of the study? Because I just wonder, is this a long-term solution, or is this something that works immediately and then potentially has no effect anymore?
I can get back to you on that. I don't remember how long it was. Okay, so thank you. I just want to thank these people. Matt Newman for finding my paper, and Catherine Mathias for helping me look over it, Mark Shackman for helping me with the whole process. Amanda Challey for my presentation review, and Sydney for getting me on my game yesterday, and Mr. D for just getting me to see the big picture, and anyone else on the journal club team who helped me. So, does anyone have any ending questions?
Okay, so next week.
Next week we have DDA presenting on muscular dystrophy. Yeah, next week too. So next week you have DDA presenting on muscular dystrophy. It's definitely going to be a great presentation, so you should all come out and see it.
I want to see this many questions next week.